right, you cool cats and kittens, good morning and welcome back to biology. All right, so last night your assignment was to watch a video about what the codon chart is and what codons are. It was only 2 minutes long, but it's right here. I wanted you guys to just kind of get used to the word codon, what their terms are, why it's important. It's hyperlinked up here if you haven't gotten a chance to watch it. It's only like 2 minutes long. So now that we're familiar with what DNA and RNA is in regards to transcription and translation, we're going to translate our own DNA using the codon chart right here on the left-hand side. This one is the circle chart, as we discussed we would be using yesterday. And then as you recognize from the video that I had you watch, there's also a rectangular one. I personally prefer the circle chart, and that's why that one's chosen here. Before we get started, I want you all to remember a few key points, and I've made sure that you have access to those key points right here in your notebooks. The first thing I want to do is bring your attention to some of the key things that we've discussed so far. So far, we recognize that DNA is used to make a copy of mRNA in a process called transcription. mRNA leaves the nucleus and goes to the ribosomes. There are 3 bases that create a codon, 1 codon that creates a single amino acid, and a chain of amino acids. Acids are a protein, and that protein synthesis, which is also where we're translating the RNA into amino acids, it's called translation, not transcription. That's why when we're translating and transcribing, they're 2 different things. Transcription is DNA into RNA, and translation is RNA into protein synthesis. To double back, yesterday we went over in your slide 17, we labeled all of the different parts of transcription all the way through translation. And just to recap, just like I just stated, DNA is right here inside the middle of the nucleus. And when DNA decides it's going to create more instructions for the rest of your body, it turns into, it creates a piece of mRNA or the messenger RNA, and that messenger RNA leaves the nucleus through a nuclear port and into the floating into the cytoplasm freely. And where that mRNA is in the cytoplasm, if you remember, yesterday I used a pencil to represent the mRNA, and then I used the tape as ribosome RNA or nRNA, and that was that little object that kind of works as a tool and goes through the mRNA, giving the tRNA, the transfer RNA, something to attach to so that it could create the proteins coming out of it. And what I wanted you to take away from this is that, and you should have written this down, your DNA to RNA is transcription, and your RNA to protein is called translation, and that proteins are made of amino acids. All right, going back to what we're working on today, I want to draw your attention to the DNA sequence that's given to you here. If you remember when we looked at Chargaff's base pairs on slide 15, we talked about the fact that with DNA is base pairs that connect together, and adenine always goes with thymine and guanine always goes with cytosine. That's just how it is. It doesn't fit any other way. It's like those puzzle pieces, and that's the only way that they fit. So A with T and G with C, and vice versa. Now, when we're talking about the when we're talking about the transcription of DNA into RNA, we're looking at switching up a few things. Back when we're looking at the DNA, we had A with T and C with G. However, when we transcribe DNA to RNA, another compound comes into play, and that compound is called uracil, which completely replaces thymine in this transcribing process, which means that when we're looking at transcribing the DNA, these codons into other codons throughout the RNA, we're looking at replacing the T's, the A's, the C's, and the G's with T with A with U, not with T, C with G, and G with C. Just like I always do, I've created text boxes within your notebook so that you can just follow along. So right now, I'd like you to take the time to pull up your notebook, make sure you're on slide number 18, and we're going to go through this first example together. After this example, you're going to go into breakout rooms with your partners, and the two of you are going to finish translating and transcribing the last two sequences. So I'll give you a minute to do that. All right, so we're going to start with this top one. I don't even want you to worry about what's going on with the codon right now. Step one is to take the DNA and you're going to transcribe the DNA into RNA, going codon to codon. So 3 to 3 to 3. In this case, we discussed that T, when transcribing, T goes with A goes with not T, but U, and C goes with G. We can move on. C goes with G, A goes with not T, but U, and T goes with A. And we'll keep going. G with C, G with C, and A with U. Then we have A with U, T with A with, or T with A. And the last codon would be A with U, C with G, and T with A. All right, now that we have our new codons for the RNA, that is when the codon chart comes into play. So if you recall from the video, when using a circle chart, we start with the middle and then we spread out. In this case, our first codon that we are going to translate now, because we're going from RNA to amino acids, is AUG. Because we're starting with A, I look for A and move to find U, and then from U, I go to G. And if you notice, G, it's got something called met start. That's kind of important. So I'd like you to insert a shape, circle met start on the chart, if it'll let me. And then I'd like you to make that circle transparent and change the border color to something that stands out, because this is kind of important. If you notice, I'm making it in red, which, as you recall, red means there's something important that's going to have to go into your key terms. So, met start. Met is actually the amino acid that we want to put here. I don't know why that is like that, so I'm going to switch it up. But met is what it translates to. AUG translates to MET. And start is kind of important because that is a typical start point when creating amino acid chains. AUG is a starting amino acid, and that's why, ding, 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 red. That means this key term, it's not the MET, but it's the start. I'd like you to put start into your key terms as a typical beginning start of the amino acid. And we'll get to that later. Going back to GUA, we start in the middle, look for the G, go to U, go to A, and if I move this over, GUA translates to val, VAL. Going over here, CCU, CCU is pro, PRO, pro. And then we go to UAA, and it says stop. Stop is important because when translating RNA to amino acids, if there is a stop protein or a stop amino acid in the chain, it stops. We don't go any further. So I'm going to italicize this and make it a different color so that I recognize that I stopped there. I'm going to tell you right now, I chose this particular chain because it had the start AUG and it had the stop. The other two amino acid chains that you are going to be translating and transcribing do not have a start and do not have a stop, only the first one. I am now going to place you in your breakout rooms with your partners, and I would like you to both work together. Somebody share their screen and work together, and in your own notebooks, I'd like you to transcribe the DNA codons and then translate the RNA into the amino acid chains using your codon chart. Any questions, comments, concerns? Let me know now in the chat or forever hold your peace. Not really. You can always just raise your hand and I'll pop into that breakout room. Thank you so much. I appreciate you all.